きまして、モレキュラープロファイルでございます。がん治療薬の開発、ドクター・ロブ・ハリス、チーフテクニカルオフィサーのお話をいたします。
Good morning, everyone. I'm Rob Harris. I'm the Chief Technical Officer for Molecular Profiles. Firstly, I'd like to provide you with a brief introduction to our company. We are a pharmaceutical development services company, and we offer a wide range of services all relating to early stage development of drug compounds. Right the way from pre-formulation through formulation development, right the way through to clinical trials manufacturing. We also have an array of sophisticated analytical technologies at our disposal that can be used for problem solving and for IP patent litigation support as well. An interesting statistic is that 30% of all new compounds in development currently are for oncology. Another interesting statistic is that 80% of all compounds in development are classified as poorly water soluble. So the link here is that the majority of compounds that require development are poorly soluble, and the vast majority, a similar proportion, are in the oncology market. So the link is obvious. There's poor solubility is linked to poor bioavailability. And what I'd like to do is just spend a few moments just giving you an indication of some of the technologies that we can provide to overcome these issues. So the system that we use in order for us to decide which strategy is most appropriate for a compound is the developability classification system. And what this does is allows us to take information on the permeability of the compound, the solubility of the compound, and also the dose requirements, and to basically categorize that in this box here. And the majority of compounds in development are considered class 2 compounds, which come into this box. And this can be further divided into compounds which illustrate or exhibit either dissolution rate limited bioavailability behavior or solubility rate limited bioavailability behavior. So effectively what this has led is to us to provide a process chain which decides which process and which technology is most suitable for the compound. We classify these technologies as enabling technologies, as you see in the box below. So what I'll do is focus to start with on compounds which can be considered dissolution rate limited. And effectively the technologies which are suitable for these compounds is particle size reduction. This basically is a reduction to, in many cases, less than 1 micrometer in particle size. So the basis on which this technology is established is the fact that the dissolution rate of the compound is directly proportional to the surface area of the drug. This leads to a smaller particle size, providing faster rate of dissolution and a more rapid drug absorption profile. This rather busy slide gives you an indication of a feasibility assessment process that we follow for a typical nanometer process. So we start off by characterizing the compound. We carry out some screening exercises to establish which is the best stabilizer for the compound to stop the nanoparticles from agglomerating. And that leads into larger scale nanomilling trials where we basically optimize the processing. And of course it's important for us to assess the dissolution characteristics and the chemical and physical stability characteristics of these compounds as well. This is an example of a project that we ran last year on an oncology drug, and it illustrates the reduction in particle size which can be achieved, and also the related increase in dissolution rate, as you can see from the slide. And this is another project for another oncology drug which we're currently working on. And this, although it's in a rat study, so it's early stages at the moment, it does illustrate the capability of this technology to significantly enhance bioavailability. And you see the link here on particle size and bioavailability. So the smaller the particle size, the greater the bioavailability. So another technology range that we also have capability to support clients with are the solubility enhancement technologies. And I'll just talk about one which we've introduced quite recently, which is melt extrusion. This technology basically provides you with a drug polymer solid dispersion which enhances the solubility characteristics of the compound. So the process, if you're not familiar with it, is simply to add the drug and a polymer into a heated barrel. And what comes out here is a combination of the drug and polymer in an amorphous glassy state. So the drug is in a monomolecular dispersion. It's effectively a solid solution of polymer. This slide just gives you just an overview of some of the polymers which are used in this technology. These are just for one company. There are other companies' polymers that can be used as well. Again, similar to the non-linear exercise, we follow a screening exercise to decide on which polymer is suitable for the compound we're working with, and also to establish the optimal conditions for processing. This slide just gives you an illustration of the solution capabilities of polymer matrices compared to the meat drug. Just to bring to your attention the fact that what I've been talking about for the few moments are production of an intermediate. And of course, you need to convert that intermediate into a conventional pharmaceutical dosage form. So this slide just illustrates the fact that you can't produce conventional capsules and tablets using these intermediates. And lastly, this is just a slide to show you that there are commercial products available that use these technologies. So it basically shows that there is a regulatory precedent for using these technologies for new compounds. And that brings me to the end of my presentation. Thank you for your attention.